Now you are what we call an organism. And as the name indicates, it means that you're made up of different organs, like the heart, the lung, or the liver. If you take a closer look at these organs, you'll see that they're made up of a mishmash of different cells that each have a very specific job to do and work together as a team. Now that is how you run a business. But cells can also make other things, like a plant, a mushroom, your house cat. And not everything that lives is made up of different cells. This is the case for what we call unicellular organisms, like microorganisms, for instance, bacteria or yeasts. This is a human cell. It contains a liquid called the cytoplasm that contains all of the molecules necessary to keep the cell up and running. At the center, there's a nucleus that contains the genetic information of the cell. Both of these compartments are filled with molecules, of which a very important subgroup called the proteins. They're sort of the workforce of the cell. Of these proteins, some have an extra layer of superpower, which allows them to break down or mend other compounds, and these are referred to as enzymes. Now let's have a closer look at what actually happens inside the nucleus. So you've probably heard of a genome, or DNA. It is the genetic information found in every single one of the cells of your body. It's sort of the blueprint of the beautiful cellular architecture. Whatever is written in there determines everything, from the size of your nose to how likely you are to get that sweet summer tan. DNA is found in the cell in a tightly aggregated form, 2 strands in the form of a double helix. When we take a closer look, we can see that it's made of only 4 colored blocks: A, T, G, and C. The way in which these blocks are positioned consecutively determine what they code for. And a series of these blocks is called the gene. It's that easy. But I have yet to explain how DNA gets to determine the behavior of the cell, and how it gets you to be so yummy brown in late August. Now, there's a little something in biology called the central dogma. It entails that when a gene is needed at a certain time, proteins and enzymes bind to it, make a copy, and turn it into protein. Let me explain. First, the DNA strands have to be pulled apart. This is done by an enzyme called helicase that cleaves these bonds, leaving room for another enzyme called a DNA polymerase. This latter enzyme makes a copy of the DNA strand, called RNA, in the process of transcription. RNA is called that way because it is similar structurally to DNA, but it is single-stranded. RNA is also different from DNA in the way that it can exit the nucleus and enter the cytoplasm. Here, it can encounter free-roaming complexes called ribosomes. These can bind to the RNA and read it sort of like a recipe. And based on the template they are given, they produce what we call polypeptide chains. These chains constitute the basic subunits of the future protein. The polypeptide chains first have to travel through the different cellular compartments and undergo modification, maturation, and folding to yield the fully functional protein. These proteins can after either stay inside the cell and do their job there, or exit the cell and exert their function elsewhere in the body. The 3-step adventure from DNA to RNA to protein is therefore called the central dogma of biology.